Okay, can you hear me and can you see the presentation? Okay. So last time we were talking about detection assays, and assay is a procedure, or assay is a test which is done to detect whether the biomolecule is active and is performing the reaction that it is supposed to perform, and to determine the titer or to determine the concentration of that particular protein or biomolecule in the sample. So assays are done, and assays are very important and very routinely done in the case of enzymes, in the case of proteins. Now, the stepwise analysis method is, we are going to do a quick revision here, is the detection, identification, concentration of a biomolecule component of the cell is only possible because of the stepwise analysis method. So there are several methods, and the sequence of those methods that needs to be done for detection, identification, and concentration, there is a particular sequence. So the biomolecules will be subjected to this through this sequence, and then finally you get to know the entire profile of the biomolecule. Now, when the biomolecules, we talk about biomolecules, there are complex biomolecules which we are referring to as proteins and say some polysaccharides. But in the analytical or in the organic chemistry, there were some techniques that were already development, some simple tests that were done to determine say basic components or simpler molecules like sugars, amino acids. So there are these very simple tests which are done to determine these simple biomolecules. But these are the building blocks. These make a big complex macromolecule. Okay. Now, analysis and characterization of these complex macromolecules is difficult as compared to the analysis of simpler biomolecules. So over the years, and it is not for a very long time, but just merely from the last 4 to 5 decades, there has been a lot of progress. There has been a lot of development in the techniques for purification of these complex macromolecules. Okay, complex macromolecules is a lot of stuff that we study in biochemistry. So, for example, any polysaccharide you're going to take is complex because it is made up of monosaccharides. The building block is the monosaccharide. Protein, the building block is amino acids. Then you have lipids, fatty acids, and glycerols. So they actually come together to make these macromolecules. So the development of the technique for purification and identification of these macromolecules has been developed very recently, just for the last 4 to 5 decades, and there is still a lot of upgradation happening. New techniques are still coming. So the routinely methods that are available for the purification are simple precipitation, centrifugation, gel electrophoresis, chromatography. If you look at it, they all are actually based on different properties. These are all diverse methods also. They are using different properties of the biomolecule for the purification. So these methods are diverse, and they are constantly undergoing a lot of development, and a lot of improvisation is happening in these methods. Even if they are diverse, they are still interrelated methods. Okay, they are still interrelated. They form a sequence. It's a chronological order that the particular biomolecule has to undergo when it has to be purified, it has to be studied. So let us in short, we will see about centrifugation, which is one of the methods that is used as a step where you want to actually isolate. I told you that when you are talking about any biomolecule or protein or any other cellular component, any biomolecule of your interest, it needs a source. First, you have to identify a source which produces this particular biomolecule. That could be a protein, that could be a lipid, that could be some polysaccharide, it could be some natural product that the cell produces, a natural substance which is used in making drugs or anything. So you have to first identify the source, who is producing that particular biomolecule. If it is a bacteria, then you have to identify the source. If it is a plant source, if it is an animal source, okay, so these are some sources. Or you obtain it from soil. So what is the source of your particular biomolecule that you are going to study? After you identify the source, then you work on the production of it. After you produce, after you're able to optimize the conditions, and if you have succeeded in optimizing the production medium, then you need to extract your protein. Now there are several ways how would you get the protein out of that particular mix, because as I told you that when you are producing, when you have optimized the medium, if you are working in the laboratory and you have identified your source is not going to produce only the product of your interest, it will produce a lot of other stuff. So you need to separate that also. So you need to separate the molecule of your interest from the rest of the molecules. So centrifugation is used. It works on the principle of rapid spinning. So you're going to take your sample mixture, actually you're going to spin it at a high speed, and that is the centrifugal force. This centrifugal force works on the biomolecules which is there in the solution, and the separation of the biomolecules is based on the differences in weight. Okay, so you have a centrifuge tube which looks like this, and there are different you can take different amount of it. So the tubes vary from size to size. You can take anywhere from, if you have a small tube, you can take anywhere from say 5 ml, 10 ml, 15 ml, 20 ml, depending upon how much mixture you have. If you have less than 5 ml, then you can also use Eppendorf's, which are there in the laboratory. You can use, there are two centrifuges, like one is a tabletop centrifuge, which is a small one, which is not very heavy. You can lift it from one place to the other place. And when you have small samples, like when you are working on samples which is 1 ml or even less than an ml, then you can use a centrifuge, a microfuge. It is called as a microfuge because it is working on smaller samples. Okay, small volumes of sample. When you're using larger volumes, 50 ml, 100 ml, 200 ml, okay, so when you use such larger volumes, then you use a bigger machine, because you're going to keep your sample into that machine. You're going to pour the sample into a container, and you're going to keep the container into the centrifuge. That container could be a tube, or it could be a bottle like thing also. So it has a cap. The cap is there or lid is there just to avoid the sample from coming out, because you're subjecting the sample to centrifugal force. Okay, do you know that there are two types of forces, centrifugal and centripetal? Have you heard about these forces, centrifugal force and centripetal force? Do you know what is the
Yes,
difference?
ma'am.
What is the difference between centrifugal and centripetal force? In short, if anybody can tell me, or you can write it in the chat box, is centrifugal force away from the center or is it towards the center? And what about centripetal forces? Is it away from the center or is it towards the center? Waiting for your answer in the chat box. Yes, very correct. Centrifugal force is away from the center. So you have an instrument, or if you have an equipment here, if you can look at this equipment here, you will see that this is the body. Not all of them are circular. They are like cube. The entire structure is huge, cube-like structure. It has a lid. You open it, and inside the lid there is a rotor, and in that rotor you can keep your test tubes. Test tubes in the sense, the test tubes, not the ones which are there in the laboratory, not the glass ones. There are special centrifuge tubes which look like test tubes, which are bigger than test tubes. Okay, and they're made up of high quality plastic material. They are not made up of glasses. So you pour the sample into the tube that you're going to put in the centrifuge. So different sizes of different sizes of centrifuge tubes have different rotors. Okay, So there are different rotors. So depending upon how much your sample is, you will choose the centrifuge tube. Now you pour the solution. Now this solution is the mixture of lot of stuff, like your the biomolecule of your interest, and rest of all the other junk material is also there in this. So you now add to it, you put it into the centrifuge. Now you must know that a centrifuge rotor, now if you look, the top view of the rotor is like this. So 1, 2, 3, 4. Suppose now this can hold only 4 samples. Okay, so it means that there will be many actually holders, but when you are going to keep the centrifuge tube, always balance the opposite forces. It is like a seesaw. Okay, if you keep one tube here, then you cannot start the centrifuge with the other tube kept at the neighborhood. So here you will have to keep the two tubes opposite to each other, so that you're balancing the weight. Okay, also if you are taking two tubes, you must take two tubes always. You should never start the centrifuge with one tube. Always start the centrifuge with at least two tubes, equal number of tubes. They are equal and opposite to each other. The amount of suspension that you are going to take in both of them will be the same. They will be of the same weight. Okay, if you have only one tube with your suspension, that is what you want to work with, then you have to fill the other centrifuge tube with water. Okay, that's because you're balancing the weight. So water wala test tube, you can throw it off later because that does not have your molecule of interest. This one is the molecule of your interest. So after the spinning is over, you can take your tube and then you can work on it. So always remember, if there are 4, 5, there will always be even number of holders in the centrifuge rotor. But it depends on how much suspension you have. You may just have one tube, so rest of the 5 will be empty. Okay, but it doesn't mean that you start the centrifuge with only one tube. So what do? You take another tube, you fill water into it to balance with the weight of your suspension tube, and then keep them exactly in front of each other, so that now it is balanced. If you don't balance the motor, then the motor can the speed, it's not smooth, it does not work smooth, it'll make noise and you may break the rotor. There is a shaft here below, there is a holder here below which holds the rotor. If this rotor is imbalanced, what will happen? It will just make a very loud rattling noise, and that might cause breakage here. These equipments are very, very expensive, so if they break, then they cause a lot of servicing amount. Secondly, it is also dangerous because the person who is working with it then can also get hurt if the rotor breaks, or if because due to the imbalance it just comes out, it pops out, it might also hurt. So this is something which you must avoid when you're working with a centrifuge. Now when you subject the sample to a high centrifugal speed, there will be molecules in this suspension of different densities. Okay, so different densities will have a different sedimentation rate also. What do you mean by sedimentation? That at a high speed they will all sediment down. So the denser molecules will sediment, the lighter molecules will remain in the liquid which is above. So this sediment is called as a pellet. You can also call it as a precipitate. So the molecules which are there in the suspension, they will have different densities. Different densities will have different sediment rate. That is the time required for them to form a precipitate. Okay, so based on that, you will get two phases. The first phase is the precipitate, which is at the bottom of the centrifuge tube. Then the other phase is a supernatant. The supernatant is containing of molecules, but these are of lighter densities. So then you can separate. You can now have a supernatant. You can take another test tube, pour off your supernatant, or take a pipette and aspirate the supernatant and transfer it, because if you just shake the tube, you might disturb the pellet again. Sometimes the pellet is loosely bound. It is loosely bound. It might get disturbed because of the shaking that the person who's handling causes. So remove the supernatant carefully, and then you will have a pellet to work. So depending upon your biomolecule, where is it present, you have to identify. Is the biomolecule present in the supernatant? Is it present in the pellet? If you identify that, how would you identify? How do you detect? You can do an assay. You can do an assay. You can do a gel electrophoresis of both the samples. You will see the corresponding bands because you're going to have a standard marker. You know that the protein you're working is probably 30 kilodaltons. So if you see a band which is corresponding to the 30 kilodalton molecular weight band in the protein marker, then you know that your analyte is here in this. You do the assay, you add a substrate to the protein that you have now isolated, and you incubate it, you add the reagent, stop the reaction, and you will see the development of a color, do a spectrophotometric analysis, and then you will see that yes, the enzyme or the protein or the biomolecule of your interest is there in the supernatant or in the pellet. Now I told you that these are larger volumes. So centrifuge is used for a larger volume. This is a Eppendorf. Have you seen the Eppendorf in the laboratories? I'm sure, yes, at least some of you all have seen it in the lab while doing some experiments or while doing some practicals. Have you all? Yes. So this is how a Eppendorf looks. Now this Eppendorf can be of, again, of various capacities. So 1.5 ml, 2 ml, 1 ml, okay, even smaller than this. If you have seen the PCR vials, if you have, I think in our laboratory we do have a PCR, and there will be the PCR Eppendorfs of the PCR vials also. They are extremely thin walled, okay, and they are extremely small also. So based upon what volume you are working, you can decide whether you want to use a centrifuge or you want to use an Eppendorf for smaller amounts. And so this can fit in a microfuge. The microfuge also has the same principle. There is a rotor in the microfuge. You also have to balance the microfuge. There will be a lot of holders in the microfuge. Actually, microfuge comes with about 12 or even more than that holders. So you will have to counterbalance. Okay, we will see a video where you will see this. How does this equipment look and how does it work? That'll give you a clear idea then. So based on the differences in the weight, you can separate the biomolecules. The heavy material will settle down as a pellet and the lighter material remains in the supernatant. This is one approach when you are separating the biomolecule of your interest from the other contaminants or from the other components of the cell. Then there is electrophoresis. See, now electrophoresis is different, centrifugation is different, but still they are interrelated because they are forming, they will help you in designing a strategy when you want to isolate, when you want to identify, when you want to purify your biomolecule of interest. So here what happens in the last slide, in the centrifugation slide, you saw that the weight, the difference in weight becomes the prime property on which you can separate. But here in electrophoresis, you will see that it is size and charge of the biomolecules. We have already studied electrophoresis, agarose, PAGE, 1D, SD, SDS, then 2D gel electrophoresis also we have seen. So now we are very well acquainted that size and charge of the biomolecules are the property on which the electrophoresis works. Now again, they have the biomolecules of different size will have different charges and they have different rates of migration. We know that there are two electrodes in an electrophoresis. The movement of a molecule in an electric field is known as electrophoresis. So these molecules are going to migrate. When you apply an electric field, they will migrate from one electrode to the other electrode. Based on their charge, the positively charged molecules are going to go towards the cathode. The negatively charged molecules will come towards the anode. So based on their charges, they will migrate. The different molecules, because of their different sizes, will have different rates of migration. Gel electrophoresis methods are very important. We also saw that they are used for DNA, protein. They are also used for lipids. So separation of these biomolecules, it can be used. There are certain modifications in gel electrophoresis. We saw 2D gel electrophoresis, which is two-dimensional, which works on two properties. Which are the two properties on which the 2D gel electrophoresis works? You just gave your exams, you just wrote your exams, so you must have studied what are the two properties on which the two-dimensional gel electrophoresis works on. Quickly give me the answer in the chat box, or if you want to put on the mic and answer. At least give me one property on which it works, the two-dimensional gel electrophoresis. Size and charge is the normal electrophoresis. The native gel or the native page that you do works on that. But what about two-dimensional? Isoelectric pH is the first property, and then the second one is, yes, molecular weight. Yes, correct, Sharvari. Or you could have message to everyone so they could see it is a direct message. It is molecular weight and isoelectric pH. So it's molecular weight. So what do you do here? You first run a separate isoelectric pH gel. You make a tube gel, you load your sample, you find the isoelectric pH. What is an isoelectric pH? A pH where the charge on the protein is 0. Okay, the charge on the protein is 0. It will contain equal number of positive and negative charges. So you will find out the pH of that particular biomolecule where the charge on it is 0. After that, you subject the protein to SDS. Molecular weight separation is SDS gel. So you run an SDS gel and you separate that on the basis of the molecular weight. So this is 2D gel electrophoresis. Rest, page, native, they are known as, sorry, page is native itself, sorry, SDS. Page and SDS, they are 1D. So you're only exploiting, or you're only using one property while you're running the gel. Whereas in 2D, you're using two properties. One is isoelectric pH and the other one is the molecular weight. So this is one technique. So they are interrelated. So after you do centrifugation, then you can subject the sample to electrophoresis to find out further details, that what charge does it carry, what is the molecular weight of it. Okay, you can find that out. Then after you do electrophoresis, you can opt for chromatography also. Once you have determined what the analyte is, then you would want to purify. So why chromatography? Because it is very sensitive. It is an effective technique where you can separate the components in a mixture. Okay, it is widely used. It is used for quantitative and qualitative analysis. For both, you can use chromatography. Method involves passage liquid or gaseous solution, passage of liquid or gaseous solution of the test mixture to flow through a tube or column packed with finely divided solid material. So we saw that we saw different types of column chromatography techniques also. So what do you do in column chromatography technique? You pack the column, you use a certain matrix and you pack it into the column, and then you pass the sample through the column. When you pass the sample through the column, the biomolecules will interact with the matrix. Okay, some of them will be retained on the matrix, some of them will pass through the column. The larger molecules tend to pass quickly through the column. The smaller will tend to be retained on the column, on the matrix, sorry. Then you elute them and then you process for the further studies. So this can be liquid chromatography, there is gas chromatography. Okay, there are different things we saw. Paper chromatography, we also saw paper chromatography. The tube or column packed with a fine solid material that may be coated with an with an active chemical group or an adsorbent liquid. So we also saw affinity chromatography, where you have a matrix and that matrix is then coated, or there is a ligand or an active chemical group which is attached to that matrix. And then you make the reaction between the biomolecule and the matrix very specific because of this ligand that is attached. Okay, sometimes the charge is used, so that is ion exchange chromatography. Sometimes specific reactivity is used, that becomes affinity chromatography. Sometimes the size of the molecule is used, and that becomes the gel permeation chromatography. Okay, again, different components of the mixture will separate due to their migration rates. So here also migration rates is very important, because the sample or the biomolecules will interact with the porous stationary material, and that is how you can separate. Then that is how you can concentrate the biomolecules. You can keep on running those batches, you can separate, and after you run say 2-3 batches, you will have more amount of your biomolecule, which is purified now, which is not in a mixture, it is purified. There are various chromatographic techniques that become a part of a strategy. So sometimes it is not only one column chromatography that you have to do. You might have to do first a size exclusion chromatography, then you might again subject your sample say to affinity chromatography or ion exchange chromatography. So there is not only one chromatographic separation column that you will have to run when you are working with a biomolecule. You might have to take another opt for another column chromatographic technique based on how much purity you have obtained. Okay, gel chromatography packed with porous stationary material. Now molecules will flow through the column, diffuse into the matrix, and it will be delayed. When the molecule enters into the matrix, we also saw that, see, if these are the beads, if this is how the column is packed with the beads, and if the molecule, which is small in size, enters in one, so there is a networking system that is formed. These beads are now closed, so you can have another bead in between. So the pores, so the molecule which enters inside will travel like this. So if it travels like this, it is taking a longer route as compared to the route which is directly going down. Okay, so this molecule will have to travel a longer distance because it is passing through so many beads. So it will take a long time to come out of the column. Whereas if the particle is a big one, if your biomolecule is of a larger size, if it's a larger molecule, of course this larger molecule will not be able to enter into this small pore. So it will just come down. So yes, the molecules which will flow through the matrix will take a long time to elute. So their migration will be delayed. Larger molecules will flow quickly. Okay, then there is another ultracentrifugation. Centrifugation is the basis. Ultracentrifugation is another different type of centrifugation that we will also see. So ultracentrifugation and gel electrophoresis also combined can be used, and that can be one of the method to determine the molecular weight of biomolecules. If the matrix is charged, then the separation occurs on the basis of charge, which is also called as ion exchange chromatography. Ion exchange chromatography is preferred because it gives a higher resolution in the purification of native biomolecules. It does not distort the molecule. The molecule that is retained after running or after eluted after running in ion exchange chromatography is active. Okay, then most of the enzymes which are used in biochemistry, in molecular biology, they are purified using ion exchange chromatography. After, when we have talked about a lot, about why these separation techniques are important, and focusing on protein also, most of it is applied to the study of proteins in biochemistry. That how do you plan to separate the protein of your interest from the main mixture of whatever medium you have got. Now, why do you need to isolate a protein? That is a very basic question. That what is the need to isolate the protein? Can you tell me at least one reason that why should you isolate a protein from a mixture of lot of other biomolecules. It could be any protein, any industrially important protein, or any therapeutic, any protein that is being used in a drug, a hormone, or a metabolite. It could be any, it could be a protein, it could be a biomolecule. But what is the separation? Why should you isolate? And why should you get it in a pure form? Anybody wants to answer? Why is it required in the pure form? The first is identification of the source, that you have identified that what is giving you the protein or what is giving you the biomolecule of your interest. The second comes the isolation, separation, detection. But why separation? Why actually isolating the protein? Why purifying it? Why can't you just go ahead with whatever you have got? To study the structure and function of the protein that of your interest. That is one of the reasons that you must purify the protein. Okay, then if, of course, the target protein, there will be one target protein that you are aiming to isolate. So this target protein is in a pool of thousands of other cellular and tissue proteins, okay, which you do not want. So it can be attached to some fragments of the cell, some other cell organelles, or some tissues, or thousands of other cellular components. So you need to be sure that whichever is your target protein, it is in the pure form when you want to study on it. So you have to separate them. The isolation, the reason for isolation is that to separate it or to remove all the other material apart from your target protein. Now, proteins which are there in the mix, these proteins are in various sizes. Who decides various sizes? The number of amino acids. Okay, the number of amino acids also decide what are the charge. What is the charge on the particular protein? So size may vary, charge may vary. Okay, so hence now they vary in the sequence also. So the amino acid sequence is also different. Is different, which will now determine the specific binding sites. So there is so much to study, and this is only possible when you isolate a particular protein. When you isolate your target protein, you will be able to study what is the size of it, what is the charge on it, what are the number of amino acids involved in it, what is the amino acid interaction that is happening, what is the sequence of the amino acid, and then because of the sequence of the amino acid, how is the binding site. We can study the binding sites also, the catalytic sites of the protein also. The properties mentioned above will help you now to decide that what purification technique needs to be followed. So once you know the information, once you know the properties of this protein, then you can design a more effective way of isolating it, purifying it. Because one way, see, if there are two ways of isolating it, and if in one particular method you get an active protein but you do not get more of it, the yield is less. Okay, and in the second method you get an active protein but the yield is more, so you will obviously opt for the second method. So when you run different strategies, you will also calculate what is the recovery of your purification protocol, how much protein it is giving you, of what purity it is giving you, what is the activity of that particular protein. So based on that recovery, you can choose a particular optimum method to purify. Okay, now separation steps will, again, going back to those few slides, size, physicochemical properties, then binding, the whether you should opt for affinity, whether you should go for ion exchange, all of that you have now studied, you have decided what to do. But when we talk about protein purification, there are you can divide it into two parts. So first is preparative and the second is analytical. when I talk about protein purification. When you talk about preparative protein purification, you are actually talking about producing larger quantities. Okay, in the preparative protein purification, you are talking about producing larger quantities of what? Of the purified protein. So you're talking about the yield, larger quantities, meaning the yield of the purified protein. What is the yield? So that is preparative. Example, this kind of protein purification, this kind of preparative protein purification is used for commercial enzymes that are used in industries. Now, when you talk about analytical does not require larger quantities. Small, relatively small quantities, small amount of protein. So the whole focus is to obtain some amount of protein. It does not need to be large. It needs to be a small amount of protein. Why? Because you are further going to study them. So this is for the purpose of research. So for the purpose of research, you will need a small amount of protein. So any technique that you use to obtain small amount of protein only for the research purpose, only for the study purpose, and that purification is known as analytical purification. So why is this approach, the analytical approach of protein purification, important? First, you will get a small amount where you can research on the protein, you can do further studies in the lab or small quantities, and based on this you can identify the protein. Then you can study the structure in detail, you can characterize it. Okay, if are there any post-translational modifications possible, you can study that on a smaller quantity of the protein. And then the function. So these analytical, mostly when you do in the laboratory, before you scale it up to preparative, before you give that technology or you give that method to the industry, before that, always at the lab scale, you always prefer to do analytical purification, where you get smaller quantities, where you have studied everything. Now once you get the profile, then you look for preparative purification, where now you know that if I do this kind of strategy like A, B, C, D, and then finally I'm getting a pure protein. Okay, after doing so many steps, I got pure protein. You have already researched, studied everything. Now you have all the data. Now you move to this where you can scale up. Okay, where you will still do the same steps, but these same steps will give you a larger yield. So now you focus on the larger yield of the production of the protein, because you're going to now develop a technology, or you're going to give this enzyme to some industry which is going to work on a commercial product where this enzyme is going to be a part of it. So the preparation, so preparative and analytical protein purification has its own significance based on what is the purpose you're going to do, or what is the purpose you're following it for. Now, when you prepare the sample, that is also very important. A protein will be there always, which is a target protein, which is a protein of your interest. Now, if this is an intracellular protein, then there is a different strategy. If it is an extracellular protein, then it becomes very easy. Okay, you don't have to look out for a protocol where you'll have to break the cell and remove the protein of your interest. So, but depending upon, is the protein intracellular, is the protein extracellular, is the protein fragile, is your target protein very sensitive, is it very fragile to any separation technique that you are going to use, then that is of no use. If it is not at all stable, if it is going to get destructed or destroyed when you're going to subject it for centrifugation or electrophoresis, then that is of no use then. Because The problem is that if the protein is intracellular, it is going to remain in the cell, which means that you'll have to break the cell. Now, breaking of cell is not easy. It is not like taking a scissor and cutting a paper. You're not going to, first of all, you don't, the cell is so small that you cannot do all these techniques on the cell. So it looks easy, but it is not so easy, because you're going to use different strategies now to break the cell. How will you break the cell? You might use some enzyme, okay, which will break the cell membrane, and then you get all the cell components out of the cell. Secondly, you can do repeated freezing and thawing. Okay, so you do repeated freezing and thawing. You have a cell mix, you have a mix, you freeze it, thaw it, freeze it, thaw it, freeze it, thaw it. Do a repetitive cycle of that, and then you will see that again, after that also, after freezing and thawing, immediately breaking of cell membrane does not happen. You will have to subject it to some another process which is called as sonication. What is sonication? Sonication is application of sound energy. So you take this cell, you take your preparation, you freeze it, you thaw it. After that, you subject it to sonication. Sonication is application of sound energy on the sample. Then the cell membrane is becoming weaker and weaker with every treatment. The pores of the cells are actually becoming larger and larger, and there will come a point where the cell membrane will rupture. And once it ruptures, all of the material inside the cell will come out. After sonication, then you have to grind it. There is a process which is known as homogenization. Okay, then you can add detergents, you can add lysozyme, and then the cell will break open. When the cell breaks open, the target protein will come out. Now, this target protein does not come all alone. Okay, it will not say that, okay, all of this is done because for me, they want me, so I will only go out and rest of you stay inside. That's not possible, because the cell has actually broken. Now there is no membrane. All of the material is going to come out. So the target protein is going to be in a pool of other contaminants or other cell fractions. Okay, so now you need to separate it based on its properties. So you can use centrifugation, electrophoresis, column chromatography, what all, and you can separate it. Okay, so this is the strategy building of how to proceed to purify, or how to proceed for isolation. So preparation of sample is one of the important step. Now, there is something which is, when we are talking about biochemistry, when we are talking about biotechnology, we are working with cells. Okay, so not all the times you are going to work with only proteins, but there can be some cell organelles also that you will be interested in. Can you name any cell organelles? What are the names of few cell organelles? Write it in the chat box. What are the names of few cell organelles? Yes, Diksha, Aditi. So far only Akshata, Sharvari, Diksha, Mitali, Aditi, Sham, Kothari, they have answered. Only these many students are there in the class right now. Golgi bodies, endoplasmic reticulum, correct, mitochondria is there. Yes, endoplasmic reticulum. Nucleus is there in the cell. Yeah, cell membrane is what we are going to break. Ribosomes are there, nucleus is there. Yes. So see, there are so many cell organelles in the cell. If you are aiming to study mitochondria, if your research demands the study of nucleus, it means that is mitochondria, nucleus, intracellular or extracellular? Any cell organelle we are talking about, what are they? Are they intracellular or extracellular? Type the answer in the chat box. The cell organelles that you all have mentioned right now, are they intracellular or extracellular? You can type I for intra, E for extra. What do you think? Intra, Yes, they are all intracellular. So what will you have to do? If you want to study mitochondria, will you have to break open the cell and remove the mitochondria? Will you have to break open? Yes or no? Yes, you'll have to break open the cell. You'll have to rupture the cell. Now there is a method which is known as cell fractionation, which is mentioned in your this unit also. So we are going to see what is cell fractionation. Cell fractionation is a procedure for rupturing of cells, where you rupture the cells. You rupture the cells, you break open them, you get the components out, you separate from the mixture of the cell constituents in an isotonic medium. That isotonic medium is very important because again, if there it is a hypertonic or a hypotonic, the cell organelle will damage. So you'll have to have an isotonic medium. Okay, as I told you, what will you do in cell fractionation? What will you do for rupturing the cells? There are different things I told you about: freezing and thawing. Freeze thaw. Okay, why you're doing freeze thaw? You're weakening. You want to rupture the cells by weakening the cell membrane. So freeze thaw is one of the method. After that, you subject it to sonication. Sonication is application of sound energies. Okay, after you do that, then you can homogenize. You will grind the suspension. You'll grind the cells. Okay, so after grinding, you can also add some lysozymes or detergents, and then finally the cell will break open. Okay, depending upon, the cell might also just break at this step also. It might just break at the step of sonication, or it will just become weak and break open in the first step itself. Depends upon what cell you are working. Some cells have very thick cell walls also. Okay, the content of the cell wall will also determine that how quickly the rupturing treatment is going to work on it. And in some cells it is so hard, the cell membrane is very tough, it does not break open very quickly, then you'll have to keep on doing some procedures repeatedly. So once now the cell will break, the target protein will come out, the cell debris will also be there, you have to send, now you have to separate. So cell fractionation is a very, very important technique which is used to rupture the cells. This is a useful method. Why? Because it helps in studying the structure, the chemical composition, and the function of the biomolecules. So you can study a lot, you can get a lot of detail only when you will be able to isolate that particular component from the cell. It could be a cell organelle, it could be a protein which is intracellular, anything, whichever is whatever is intracellular, for that you will have to go for this method of breaking the cell open. Okay, when the biomolecule of interest is stored inside the cell and not released, when it is intracellular, then cell fractionation is crucial. That is the only way you can proceed for purification or isolation. It is a method to separate subcellular components and isolate organelles and other subcellular components from one another. So not only and we are interested in cell organelles, we are also interested in biomolecules like proteins. Some of the biomolecules are organelle bound. Some of the biomolecules may be sticking to the mitochondria. Some of them may be sticking to the Golgi complex, okay, or endoplasmic reticulum. So these proteins, if these biomolecules are always associated with the cell organelles, then it is very important that you need to now separate that cell organelle. So when you get that cell organelle, you'll be able to separate the protein also. You can study both of them, whichever your interest depends on. Now, why do researchers use cell fractionation? It is a multi-step procedure that subjects the cell to centrifugal force. Again, here also you will see the application of centrifugal force. It's a device that spins. There is a motor inside, there's a rotor, and then that spins at a particular speed. Okay, because the speed and the density of the cell organelles or the biomolecule vary from each other. So based on their densities, different speeds are applied, and then you can separate them. So the denser molecules, the denser components will settle down as a pellet. The lighter one will remain in the supernatant. Okay, so the purpose of the cell fractionation is to isolate the organelles within a cell so the scientists can study them individually. What is the principle here? The principle of this analysis is that each cellular fraction is unique because they have unique properties, they have a unique structure. Okay, it's a unique or a combination of unique enzyme activities which can be assessed and used to follow purification of a particular organelle or membrane fraction. So if you know what is it made up of, what is the uniqueness, what are the enzymes which are constituting, what are the reactions that are going on in this particular component or in this particular fraction, then you can design a purification technique, you can design or you can follow a particular strategy based on more and more information that you get about that particular cell fraction. So this is how the cell fractionation actually, this is one of the diagrammatic representation. So this is the first step. You have a tissue here in the tube. Okay, these are the cells. If you can see, it's in the fine structure, it's actually a cell. 100 times or 100 G. Now G is the acceleration due to gravity. What is the value of G? What is the value of G? What is the value of G? 9.8. Yes, 9.8 watt. What is the unit that is used? Yeah, meter per second square. Very good. So it means that this sample will be now spun at 1000 G. So it is 1000 times more than G. Okay, which is the acceleration due to gravity. So at 1000 G, when it is, so first what you do is first you rupture the cell, you homogenize the cell. After you homogenize the cell, you get a homogenate. This is called as a homogenate. Now this homogenate is going to have so many different things. It is just not one particular protein, because now you have identified the source. Okay, you developed a method to get the protein out, whichever is the most convenient. If it is intracellular, then you will have to rupture. Okay, now with the first step, what you do is you're going to apply it for freeze thaw, sonication, maybe adding detergent, enzyme, grinding. So everything will lead to one preparation which is called as homogenate. So after you get the homogenate, this homogenate is a mixture of the cell debris, the broken cell membrane, your mitochondria, everything that is there in the cell has released now in the homogenate. It's a mixture, and then that is now subjected, this homogenate is subjected to differential centrifugation. Okay, in a differential centrifugation, what happens is bigger and heavier components will move to the bottom of the test tube. The lighter components will be suspended in the supernatant. So when you apply 10,000 G to this homogenate for 10 minutes, what will happen? The heaviest at this particular 10,000 G, whichever is the heaviest organelle or whichever is the heaviest component, will settle down as a pellet. So this is the pellet at 10,000. Now you get two different phases. One is the supernatant, one is the pellet. Then what you do, you take the supernatant, okay, very carefully, very gently, because you're working with cell organelles. They are very delicate. You do not want to distort them. So you take the supernatant in another test tube now. And after that, if you are interested in the nucleus, you can very well be happy that you have achieved the isolation of the nucleus. Now you can start working on the nucleus. But if nucleus is not that organelle which you are interested in, the organelle that you are interested in is in the supernatant, you have to take the supernatant and you have to now subject it to further centrifugation. What will you do? Now you will increase the speed. It was 1000 G, now it is 20,000 G, 1000 G for 10 minutes, but this is 20,000 G for 20 minutes. After you spin it for this much time at this much speed, again you get a protein, again you get a pellet. This pellet is now mitochondria, okay? Or it can be chloroplasts if the cells are from a plant. So then if you're interested in a mitochondrial study, then you can take this pellet and start your study. But again, if you are working on microsomes, the microsomes are here in the supernatant. So you take the supernatant, now you suspend it, you spin it at 80,000 G for 60 minutes, and again you get a pellet. Now this pellet will be the microsomes. So this microsome you take for your study. There will be certain more cell organelles in the supernatant. You again spin it for 150,000 G for 3 hours and you will get ribosomes. So depending upon what your target cell organelle is or what your target protein is, you will have to apply a centrifugal speed. Now it is all established. It is all there in the literature that at what speed for how much time will you get a nucleus, at what speed at how much time you get mitochondria, at what speed and at how much time you get microsomes, lysosomes and all the other stuff. So you just have to follow a established protocol and you can get, for every cell there is a lot of research done. You can just refer to some papers and you can find out a differential centrifugation process, and you can now separate them from each other. Okay, so after you have separated them from each other, you can now wash them. So because see, this pellet that is formed every time you get a pellet, this pellet is not exactly pure. It will have some components from the cell fraction which is attached to it. So you need to give gentle washings to it. You have to make sure that you're washing everything out, and then you start working on your interest on the biomolecule or on the cell organelle of your interest, and get more and more information about it. Okay, so homogenization is the first important step. Subjecting the cell homogenate to centrifugation at various speeds is very important. Now the term comes as sedimentation rate. Sedimentation rate is the time required. You can say the time required, the speed required for a particular component to settle, to pellet out. Okay, this is very, very important. So this has already been now standardized, sedimentation rate. So you will have a lot of information on what is the sedimentation rate of mitochondria or microsomes. So you just have to refer and you have to go for that particular protocol. So differential centrifugation is important. What it does is it uses different angular motions. Okay, different angular motion to separate cell organelles based on their densities. So here the property that you're going to use is the property of density. The denser a certain cell component is, the it will settle down. The lighter or the lower the cell component, lower density the cell component is, it will end up being in the supernatant. Okay, so the upper fraction and the lower fraction. The upper fraction is the supernatant, the lower fraction is the pellet. Again, we will just revise. You all have not understood the first, but now pay attention. This is your cell suspension. Whatever the interested biomolecule, whatever the biomolecule or whatever cell organelle you are interested in, now you have to extract it from inside the cell. So you're going to disrupt the cell, you're going to rupture the cell. There are series of treatments that you can give. Freeze thaw, sonication. So you take this homogenizer, this is the homogenizer. You have to spin it inside. Now you just don't spin it like this, like you have to add some beads to it, some glass beads to it. So those glass beads will create more friction, and when they create more friction, the cells will break open. So freeze thaw, sonication, homogenization, and at last you can also add some enzyme or some detergent, and then you can break open the cell. So when you break open the cell, you see that the cell membrane is disappeared. Now the cell homogenate will only contain the cytoplasm and all the other cell organelles. Now this is called as the cell homogenate. This cell homogenate now is spun at low speed. When you spin it at low speed, whatever is heavier will settle first. So this pellet that you get at a low speed is basically the whole cells, the cells which have not ruptured. There will be some remaining like that which have not ruptured. Nuclei, if the cell has ruptured, then the nucleus has also come out. So at a low speed, you will get nuclei. Cytoskeleton, we saw that the speed is 1000 G for 10 minutes. Okay, that is the low speed. After that, you remove the pellet. You start working on your pellet if you are interested in nucleus. If not, then you take the supernatant and again you subject it to centrifugation. But this time the speed is medium. From low speed, you are moving to medium speed, which is 20,000 G for 20 minutes. Okay, then you get a pellet. Take the pellet out. You will, if you are interested in mitochondria at this particular speed for this much time, you get mitochondria. Then you subject the supernatant for high speed if your interest is in microsomes. And then you the speed is here 80,000, I think, if I'm not wrong, and this goes for some 1 hour or something. And then you get microsomes. Subject the supernatant again at a very high speed, so that is for 150,000 G and for 3 hours. And then you get a pellet which is containing ribosomes. Okay, now you can wash the pellet, and then you can start the further studies. Okay, so you have to be careful that you are not damaging the cell organelles. You're not, while separating the pellet and the supernatant, you are not be shaking the tube very vigorously. So because the pellet might also dislodge. I told you some pellets are loosely bound, so if they dislodge, then they come up. If you shake this, if you stir this, then this pellet and the supernatant gets mixed, then the whole purpose of centrifugation is defeated. So you have to be gentle. Washings also have to be gently, because you're working with cell organelles. You don't have to damage them. If you damage them, you will not be able to study them properly then. Okay, so at every step on the way, assay is also conducted. If you're working on a particular protein, that is, you are isolating from the cell fraction, at every step an assay is done. What do you do in that assay? You add a substrate. If the protein is active, the substrate will go and bind in the catalytic site. You will see that it has formed a product when you read it spectrophotometrically. But if the protein at any step is damaged, it becomes inactive, then you will not see the product formation. So that tells you that this step has damaged your protein. So at every step, plus it is not only about the activity, it is also about the concentration, because when you do an assay, you also have a calculation to do, which will tell you what is the concentration. So at every step, the activity and the concentration of the protein is checked. And if it is good, then you are on the right path. So one of the fractions during centrifugation will usually have a high protein activity. Okay, when you're doing differential centrifugation, or if you're doing centrifugation, there will be one fraction which will always have high protein activity value. So this particular fraction will serve as the protein source. So now this is the fraction that you are interested in, because it is giving you high concentration, high activity, and then you can isolate that particular fraction and then go ahead with the variety of purification steps. Okay, so differential centrifugation that we just spoke is a type of, ultracentrifugation technique which comes under preparative. Preparative is getting something in a larger yield. Okay, so differential centrifugation can help you to get larger yields. It is commonly used in microbiology and cytology, the group that studies in about cell structure, cell organelles, cell functions, and all that. Cytology, that is the science. Microbiology, yeah, you all know. What is the purpose of doing this? Is to separate certain cell organelles from a cell to study and analyze the parts of the cell. Here you homogenize, you break open the cell, you obtain the cellular components, then you form a homogenate. Okay, after the homogenate is formed, you subject to different centrifugation speeds, and based on the densities, you will see that you are obtaining a pellet. Every component organelle has a different sedimentation rate. That is time that is required for complete sedimentation. So if a complete pellet, all the component, all a specific component is sedimenting, is pelleting out at a particular time, at a particular speed, that will become your sedimentation rate. For instance, when cell homogenate of cell organelles is centrifuged for 15 minutes at 1000 G, the cell organelles like mitochondria and lysosomes pellet out. They are the heavier, they are the denser, so they will pellet out first. The next strategy is to separate the tissue homogenate into various cell organelles and centrifugally divide the homogenate into number of fractions by increasing the centrifugal field at each step. So we saw that there is stepwise increase. First low speed, medium speed, high speed, and then very high speed. The centrifugal speed should be such that a particular target organelle will sediment at a known time to give pellet. So that time is very important. So if you remember, for ribosomes, it was 150,000 G for 3 hours. So that 3 hours is very important because we are talking about the rate, the time factor here. So if you exceed beyond that, then you will not get ribosomes. You will get something else. So at 3 hours, you should stop so that you will get ribosomes. So this is all known. This is all recorded now. The pellet and supernatant are separated in each step, and the supernatant will be subjected to recentrifugation so that the other cellular components will now sediment. Okay. Gentle washing of the pellet at every step is necessary to remove the contaminants. Any doubts so far in this? Anything that you all have not understood? Yes, no? So the advantages and the disadvantages, the most common and widely used method is cellular fractionation, cell fractionation, because your interest is in the intracellular organelles or intracellular proteins. And this is the only way you can get them out. It is very convenient and an economic way, but the disadvantages are that the yield is poor and the preparations are never absolutely pure because they will always have some or the other contaminant or cellular component clinged to it. So when you're working with, say, cells, when you're working with cell organelles, how will you identify? You will have to do microscopy at every step. Okay, every step you do microscopy to identify what the pellet is. If you're working on this, for example, go back to the slide and yes. So for the first cells are homogenized in the blender, breaking them up and all of that happens. You get the pellets. So the researchers use microscopy to identify the organelles in each pellet. Okay, so that is what will tell them whether the organelles are intact. Because here, if you start doing assay, the biochemical assay may not work for cell organelles. Biochemical assays work for protein, they work for enzymes, but they don't work for cell organelles. Cell organelles, you will have to do microscopy. And that is going to tell you whether the pellet has the organelle of your interest or not. Or do you need to do further more separations based on the centrifugal speed. Okay. Assays will also come because again, what happens is when you get a purified cell organelle, then what you do is they do particular reactions. So there are particular reactions that will happen in a particular cell organelle. So you can use those reactions to find out now whether the mitochondria that you have isolated is in an active form or not. Okay. So this was about cell fractionation. Now we can move to protein purification, a little more about protein purification, and from the next class, then we will focus on centrifugation, the details of centrifugation. As I again emphasize, first find the source of your protein. Okay, once you have identified the source, let me write it down because I don't have the slides here. We've already done so many slides now. This is just kind of a repetition. So identification of the source. After you identify the source, you produce it. Now you have identified in which fraction. If you're working with cells, differential centrifugation is also useful. If you're working with proteins, again, differential centrifugation also can be used. So it means that you will have to identify a fraction where you get good concentration of protein and an active protein. So identify fraction with active protein and high concentration. So you have done that now. Okay. Now what will you do? So now you subject to dialysis. So dialysis is a process where the protein is separated from smaller molecules, separation of protein from smaller molecules. Molecules and ions. So what happens here is, I told you that it's a semi-permeable membrane. A dialysis bag is a semi-permeable membrane, and you activate that. You open the pores first, and you add your protein mix. Now it's a mixture of the protein plus the other stuff. It has a cutoff value, and it will retain your protein inside the bag, but the other cellular components or other contaminants which you do not want, other smaller molecules, they will come out. Okay, it is done in a buffer system. It will all come out in the buffer, and whatever remains in the dialysis bag is the protein plus the other larger molecules. Okay, so the semi-permeable membrane is known as a dialysis bag. Then the second, after you finish, you might opt for salting out. So when you are doing salting out, this is basically you are precipitating. So when you add salt, you will precipitate. Okay. When you have a bulk protein purification, like if you have a large amount of material that you large amount of say medium from which you have to now take your protein out, isolate your protein out. For the larger bulk protein purification, there are certain different ways that you go about. So first, very commonly used is the salting out. This is a very common step. It is used to isolate proteins. Okay, so you can use dialysis if you're working on, say, smaller samples. So dialysis helps you. But if you have bulk to be from where you are going to isolate your protein, so there is precipitation that you must do, and this is a very common step. Usually what salt is used is ammonium sulfate. Okay, so I have this bacteria, I inoculate that into the medium. I have like these flasks. Suppose I have hundreds of such flasks. Okay, so finally I know that it is an extracellular protein. Everything, all the protein is given out into the flask. Okay, into the medium. What I have to do is pool all my flasks. So I have 100 of them. Each flask has about 100 ml of medium. So if each has 100, the hundreds will have, okay, this much. Now for this much, will I be able to use dialysis? Out of question. Because now first, dialysis bags are used for smaller concentrations or smaller amounts. So what do I do now? I pool all of that and I add ammonium sulfate salt to this. When I add ammonium sulfate, because of the ammonium sulfate addition, the protein precipitates. Now I take that precipitated protein. The protein of my interest will not be too much. It will be lesser. This broth, the medium, the other components, they also add to the volume or to the concentration. So when I add ammonium sulfate, the protein precipitates. Along with the protein, the rest of the things will also precipitate. But at least now I am only concentrating on the protein, not on the supernatant part. Okay, so what I do is I add ammonium sulfate. Now why ammonium sulfate? Because ammonium sulfate is highly soluble. It's highly soluble in water, because you are going to make the microbial medium, the medium for the microorganisms. It is all water based medium. So this is highly soluble in water. It does not harm the protein. It does not harm protein. It does not denature the protein. Again, what is one more important thing is whatever protein precipitates, the protein precipitates in native state, okay, in the original state. So no loss in the conformations or activity is also retained. And you can, after you do ammonium sulfate precipitation, you can take that protein for structural studies also, because the native state is not distorted here. Okay, because structural studies are done only on intact proteins, not on broken proteins, not on distorted proteins. So you do ammonium sulfate precipitation. So salting out is using salt concentration to cause precipitation. Then after you do the salting out process, you have got a precipitate now. Then you can use that for subject that to dialysis. Okay. Now, because when you have added salt into it, there will be ions into your precipitate. Obviously, when you have added ammonium sulfate directly to the broth itself, to the growth medium itself, there will be so many other things also which have precipitated. So now you want to make sure that only the protein is there, but dialysis does not assure you complete purification. It is partial purification. What it will do is, when you do dialysis, you choose a semi-permeable membrane. Now that semi-permeable membrane comes with a cutoff value. I told you that the pores, the semi-permeable membrane will have pores. Now, if you are working with a protein which is say 60 kilodaltons, okay, so you have to choose a bag. You have to choose a dialysis bag whose cutoff value, what should be the cutoff value? If I want to retain the protein in the dialysis bag, okay, if I want to retain the protein in the dialysis bag, then should the cutoff value of the dialysis bag be greater than the protein size or lesser than the protein size? So my protein is of 60 kilodalton and I want to retain it in the dialysis bag. So what should be the cutoff value of the dialysis bag? If I want to keep the protein inside, should it be more than the protein cutoff value or should it be lesser than the protein's molecular weight? You can type your answers in the chat box. So imagine a sieve. Imagine a sieve. If you are going to filter something from that sieve, if you want to retain something above the sieve, so the size of the sieve, should it be smaller than that material or should it be larger than that material? Or imagine straining a cup of tea. So what happens when you strain a cup of tea? Yes, so the size of the sieve should be smaller than the material that you want to retain. So obviously what happens is the pore size of the dialysis bag is smaller than 60 kilodalton. Okay, so it means it will remove molecules lesser than 60 but will retain 60 kDa or more. So it will retain the protein of your interest also, plus other components. I do not know whether the other components are protein or not. They can be anything, but most likely proteins. So it will retain the molecule of my interest plus the others also which are greater than 60. Okay, but lesser than 60, everything will come out. So that is what is dialysis. So when you treat the bulk protein preparation with ammonium sulfate, you get a precipitate. Now you should not keep the ammonium sulfate in the precipitate. You should have to clean the ammonium sulfate. So how do you clean it? You do it by dialysis. So you subject the precipitate for dialysis in order to remove the ammonium sulfate. So it is done using a membrane. The membrane is semi-permeable. It has pores. Now the protein is placed into that membrane. Based on the size of the protein, the membrane, you can choose the membrane. Okay. This will not allow the protein to pass out, but smaller ions, smaller molecules, unwanted, will easily pass. Okay, dialysis, the principle on which dialysis works is osmosis. The salt ions which are there, now in this case you have added ammonium sulfate, the salt ions which are there will pass out into the outside buffer and it will stop when an equilibrium is attained. Now the protein which is in the membrane inside the bag is the overall concentrated protein which does not have salt in it now. Okay, so this is in short about dialysis